Hey guys, we are going to start the section 2 of notes, which is on page 5 of your packet. I'm going to keep looking at the packet to make sure I'm giving you all the information you need to get all of your notes. So here's a great resource. Feel free to use this on Facebook when you're getting into fights with people online. But this shows you your neutral or least biased news sources versus your most biased news sources. So if your friends, uncle, whoever are posting things from NPR, BBC, USA Today, Washington Post, those are probably fairly accurate places. But if you are posting from Occupy Democrats or Breitbart, you might want to take those things with a grain of salt because those are definitely a little biased. So I'm not going to walk you through this whole thing, but feel free to look at this. It's kind of cool. So, scientific method. You've done this before. You will need to know how to write a good experiment with all of the parts of the scientific method. So we will definitely be spending a lot of time on this whole year. So we want to start off the year right. I love this little cartoon off on the side. I wonder if that happens every time. Parker's currently doing this with dropping food off of his high chair because maybe it won't drop one of the time. So he has to try a lot to see how gravity works. Horoscope activity we're going to do in class. So stay tuned for that. If you are not in class, check the beginning of the week video and I will tell you what you guys need to do. All right, the steps of the scientific method. So go ahead and pause this and write down whatever steps that you can remember. Go. Okay, unpause. And here we go. So you want to make observations. You want to do your background research or ask a question. Usually ask a question and then do your background research. Form some type of hypothesis. If I do this, then this will happen. Design your experiment. Gather and present your data. And then analyze your data and draw conclusions. Let's move me up a little bit. And then submit for peer review. And that's the big one. Make sure that people are evaluating your experiment. Make sure it can be done over and over and over and over again with the exact same results. And usually what happens is you draw your conclusions and you form a new hypothesis, you design a new experiment, and you do all of this until you finally get around to a good concrete conclusion. So there we go with that. So making observations. An observation is any objective fact learned by using human senses. So things you can see, things you can hear, things you can taste, things you can touch, things you can, what is the one I'm forgetting? Oh my goodness, I need a brain. Any of your senses. An inference is a reasonable conclusion based on observation and evidence. So most of our observations are going to be by sight, but you can use one of the other ones. Hearing, I think is the one I forgot. So these are more subjective. I can see a red shirt. I infer they are a South Florida fan. Those are two different things. So let's see what you got. There's an axe next to the bloody dead body. Observation or inference? This is something you see, so this is an observation. The axe was the murder weapon. That is an inference. You know that axes can be used to cause bloody dead bodies, but not necessarily this one. We don't know. That's an inference. The axe at the crime scene was found 10 meters to the left of the body. That is an observation. The killer was left-handed and threw the axe to the left. That's an inference. So, observation is something you use with your senses, and then your inference is something you use your senses and your prior knowledge to come to some type of a conclusion. On your scientific thinking packet, there is a separate article you're going to read comparing inductive and deductive reasoning. So it's a super quick article, and that is on Canvas. So you want to read that and answer those questions. And then here's this little cartoon, and we're going to look at making up some observations and some inferences. So the little fish are saying my word. That one came just too close for comfort, if you ask me. So think of observations you could make from this and think of inferences you could make for this. And feel free to come talk to me about those two things. And this is where humor comes in, because observations aren't funny, but inferences are funny. So the whole reason that this is entertaining at all is because of the inferences you guys are making. A man died of thirst in the desert is not funny. This cartoon, kind of funny. So anyway, that is on your packet. So feel free to fill that out as you want to. So we have our background research. Did I not do? Ooh. So background research, ask a question. So you make your observations, you figure out what is known about the topic, and you build on that. What do we still need to know? Okay, I know this. I want to know this. And that's where you're going to base your hypothesis on. So here is your testable prediction. There are 4 things, and this is in your note packet. You might want to write this down. Hypothesis is written as a statement, not a question. If I do this, then this will happen. I think or I predict is not found in a hypothesis. I think this drug will be really good for blood pressure. No. If I give this drug, then the blood pressure will come down in our patients. A hypothesis is testable. You can't. I hypothesize that I could fly on a unicorn over the mountains in Mars. We can't really test that with current funding. So, and access to unicorns. So not really testable. And then for a hypothesis to be properly supported through evidence or rejected, so those are your 2 options. You can support or reject a hypothesis. You don't get a hypothesis wrong. It is supported or it is rejected. It has to take a specific stance. You can't say, it's this or it's that. So these are our 4 options or 4 descriptors for what makes a good hypothesis. So what's wrong with these guys? Chocolate may or may not cause pimples. Kind of wishy-washy. You got to pick one or the other. I think salt in soil causes plants to grow slowly. Yeah, you're correct. However, you don't want to say think. Salt in soil causes plants to grow slowly. That is a hypothesis. There are ghosts in my closet. If you can test that, that would be awesome. We need to get Sam and Dean Winchester in here, but other than that, probably can't test that one. Is plant growth affected by the color of the light? That is a question. That is not a hypothesis. Bacterial growth might be affected by temperature, or it might not. We don't know. So make sure you're making an actual statement. Stand by your thoughts. And sparrows are happier birds than eagles. I'm not quite sure how you're going to test that one, so let's make that a bad hypothesis as well. How about this one? Is this a well-written hypothesis? So what will happen if we mix oil and water? It will not blow up. It is a statement, not a question. So we got that one. It's not wishy-washy. Oops, going the wrong way. You can test it. And they're definitely there's not I think or I predict. They're just telling us what's going to happen. So this is actually a fairly decent written hypothesis. I might state it as, if I mix oil and water, then it will not blow up. But other than that, it's pretty good. In your packet, there are some vocab that you should probably know the answers to. Here are the answers. If you want to double check yourself, I would try this yourself first to test your knowledge and then go ahead and fill in the correct answers. But these are all words you are going to need to know really, 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 really well, especially things like control group and hypothesis, independent and dependent variable, experiment. All of these you're going to need to know. These will be multiple choice questions. There will definitely be an FRQ that has to deal with these concepts. You have to know these words. So there you go. Learn them. Know them. Okay, a null hypothesis. This is on your notes in that little scroll place. A null hypothesis predicts no relationship between 2 phenomena or no difference among groups. So sometimes you want to show a relationship, but sometimes you want to say, okay, you can't always prove that, but you might be able to prove a null hypothesis. So tobacco scientists are like, oh, there's no relationship between smoking and cancer. Yeah, there is. So that is a null hypothesis. Sometimes when you're doing an experiment, you can prove or disprove a null hypothesis instead. So designing an experiment. You always need a control group. So you need an independent variable, and that's what's going to be changed. So let's say we're testing a medication. the amounts of the medication or the fact that there's a medication at all is going to be the independent variable. The dependent variable is what we are going to measure. So, all right, are we measuring blood pressure? Are we measuring whatever the drug is supposed to do? So that is what we're measuring. And then you want to have controlled variables and constants. So everything else has to stay the same. So we have people of a certain health, we have people of a certain age, we have people of a certain sex. More drugs than not are tested on males. So females, we get fun and exciting side effects when we start taking these drugs because not always are females involved in the testing part of the drugs. Yay for being female. So mix dry is something I would keep in mind, and this is on your packet as well to fill in. So mix stands for manipulated, independent, and x-axis. So your manipulated variable is your independent variable, and when you're graphing this, this is going to go on the x-axis. There's only one, but you can have levels, like 10 milligrams of a drug, 20 milligrams of a drug, 30 milligrams of a drug. Dry stands for dependent, responsive, and y-axis. So your dependent variable is the one that's responsive. Whatever you're measuring is the response to whatever your experimental treatment is. And this will be graphed on the y-axis. You can have more than one. They can be qualitative or quantitative or both. You got lots of options for these guys. So common issues with experimental design. This is also in your notes. You want to make sure you don't have a sample size that is too small. If it is not an accurate representation of a population, you can't really use that because it's not accurate. If there's no trials that have been repeated, you don't know if it's going to happen more than once. If there are uncontrolled variables in play, once again, it's not a very good experiment. You want to make sure there is a control group, and you want to make sure there's only one independent variable. Once you get more than one independent variable, it is a bad experiment. So I could see a question on a test saying, what is wrong with this experiment? And it's one of these 5 things. So make sure you know what these things are, and you make sure that you know that these are not good things to have in an experiment. So you want to make sure you have the opposite of all of these in any experiments you would design. Ideas are tested by experiment. This is the core of science, and everything else is bookkeeping. So Lois wants to find out whether feeding babies mashed carrots improves their vision. She borrows 50 babies from her son Stewie's daycare. 25 of them are given a normal diet, and 25 are given mashed carrots as their evening meal every night. That's going to be a mess. She then gives them a vision test. So what is the independent variable? What is our experimental factor? The diet of the babies, whether they have carrots or no carrots. Our dependent is what we are measuring. So what are we measuring? The vision of the babies. Maybe score on a vision test. I don't know how old babies are going to be on answering questions. Control group is what does not get the experimental factor. That's going to be the diet with no carrots. Experimental group is the diet that has carrots. And then our constants, you can look at the rest of the diet is all the same, the age, the sex of the babies. Everyone has a decent baseline vision before you start doing this, the amount of sleep the babies get, make sure all the same test is given, things like that. So you want to list some constants so the person grading your test knows, okay, you know that there needs to be constants. Then you're going to gather and present your data, usually in a data table. Qualitative data is descriptives, qualities, color, smell, texture, sounds, any of those things. Quantitative data is numbers. So how many of something, the length of something, the weight of something, how much is left, any of those things. So sometimes you have words, sometimes you have numbers, sometimes you have both. But then you would put that into some type of a data table. This is what a data table looks like, in case you weren't sure. You want to make sure you're presenting your information in an understandable format. So what kind of graph do we want to do? So discrete data is you have very specific groups or categories, things like gender, colors, ranks, very specific things. So you could do parts of a whole, percentage fractions, you can use a pie chart for. So a day in the life of a goldfish alternates between, hey, look at that plastic castle, flap our fins. Wow, someone put a plastic castle in here. That's a really neat plastic castle. Closed mouth, and then open mouth. This is the life of a goldfish. So you have it nicely divided. So you can do your pie chart. If you're comparing different things, you can do a bar chart. I'm sure once again on social media, you're seeing a lot of these right now. So there's your bar graph. If you have a measurement or ranges in continuous data, you might want to do some type of graph like this. You can compare data in a histogram. You can use just a line graph, which is going to be probably most of what we do in here. When you're 5, you know a lot about dinosaurs. By the time you're 25, you know a lot less about dinosaurs. Find a 5-year-old, they will tell you everything you ever needed to know about dinosaurs. You can do determine correlation between variables and find a trend between all the data points. So you can do a scatter plot graph. Here's another scatter plot for you. And then you want to interpret your results and see what you learn from the experiment, and do your results support your hypothesis. Never put my hypothesis was wrong. Say my hypothesis was supported or my hypothesis was rejected. And rejected is absolutely fine, because then we know what it's not, and then we can try something else. And finding out what it's not is almost as important as finding out what it is, because it's one less thing that we can worry about. So that's what they're doing with all the COVID testing. With the vaccine, okay, if this vaccine doesn't work, okay, let's find one that does work. Writing your conclusion. Restate the problem question, which explains the reason for doing the lab. Was the hypothesis supported, not supported, or inconclusive? Maybe you don't have enough information and you need more. So give us the why for that. What did the data say? What was the control group? What was the independent variable? What was the dependent? Describe the graph and any trends that are visible. All of that goes in your conclusion. Oops, sorry. Talk about any possible causes of error. Everyone has causes of error, often human error. I think every lab I ever did in college, I listed human error as one of our causes of error. What are some real-world applications of this experiment? What are new questions that have come up as a result of the experiment? And what are suggestions for further research? And then your final summary statement, which is going to answer whatever your original question is that you were trying to address with your hypothesis. Never use I, we, us, me, my, our, etc., any pronouns. So you're not going to say, we conducted this experiment over 4 days. It should be, this experiment was conducted over 4 days. Are you going to submit for peer review? So other people should be able to do your exact same experiment and come up with the exact same results. And if they don't, it's a crappy experiment. The example I give is a recipe. If you follow the recipe for chocolate chip cookies, you will get chocolate chip cookies every time. That is a good experiment. If you follow the directions for chocolate chip cookies and you get banana bread, something went wrong. So you want to make sure that you are doing it. If you're following the directions, you should get the same thing. Scientific theory. A widely expected explanation for something, and it is only formed if the experiment is repeated many times by many people and the results are always consistent. So once again, you can think of recipes as a good theory. Things like gravity and evolution are scientific theories. Theory with a capital T, the way we use it in science, is as close to fact as we can get. But the cool thing about science is as soon as we get something that disproves it, we get to change our mind. All right, you guys survived. So I want to remind you all that you are smart, you are special, you are loved, and if I don't see you, have a good afternoon, have a good evening, and good God, do your homework.